Hello, my name is Miwa, and today I'll be talking about cartilage. Before actually getting into the topic, let me explain how the highlights in my slides work. Red highlights are for terminology that you must know for the exam. Orange is for terms recommended to know for acing the exam, though not essential. And green is for terms that you do not need to memorize, but just good to keep in mind. All right, let's get started. Just like other tissues, cartilage is made up of cells and the extracellular matrix, or ECM. The cells in cartilage are called chondrocytes, and they sit in spaces known as lacunae. In the ECM, we find fibrils, mostly made of type 2 collagen, and glycosaminoglycans or GAGs, which are mostly hyaluronan. Hyaluronan is special because it can hold a lot of water. The ECM also contains proteoglycans like aggregan and multi-adhesive glycoproteins like chondronectin and fibronectin, which help the cells adhere to the lacunae. Common features of three types of cartilage. Cartilage is avascular and it's very hydrophilic due to hyaluronan. This water content helps nutrients diffuse through the cartilage despite this avascularity. Cartilage is basophilic, even though collagen itself is acidophilic. This happens because of the large amount of sulfated GAGs. Out of the three types of cartilages, two, except for fibrocartilage, own a layer called the perichondrium. The perichondrium is responsible for apodictinal growth and is eosinophilic. Its tissue type is dense irregular connective tissue, which is vascularized. You'll find perichondrium around hyaline and elastic cartilage, but not in joint areas because it has some nerve supply. Perichondrium has two layers, an inner chondrogenic layer that contains chondroblasts, which are precursors of chondrocytes, and an outer fibrous layer that contains fibroblasts. Functions of cartilage include to bear stress, to absorb shocks, to allow surfaces to slide over each other, endochondral ossification, and to provide nutrients to surrounding tissues. Now let's talk about the three different types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. First, hyaline cartilage. Hyaline cartilage contains structures called isogenous groups, which are groups of up to 8 chondrocytes that derive from the same mesenchyme. The area around each lacuna is sometimes called capsular matrix. The area around each isogenous group is the territorial matrix, which is very basophilic because of the sulfated GAGs. Between the isogenous groups is the interterritorial matrix, which is less basophilic because it contains some more collagen. Where is hyaline cartilage found? Examples include joints, the walls of large respiratory tracts, the nose, costal cartilage, and parts of the larynx, especially the thyroid, cricoid, and the inferior part of the arytenoid cartilage. Next, elastic cartilage. In elastic cartilage, the ECM contains elastic fibers in addition to the usual components. For a review, elastic fibers are stained by orsain, aldehyde fuchsin, and resorcin fuchsin. It does contain isogenous groups, but there are fewer of them compared to hyaline cartilage. Where do we find elastic cartilage? Examples include the epiglottis, the covering of the eustachian tube, and the auricle, and some smaller cartilages in larynx, like treticile, cuneiform, and the superior part of the arytenoid cartilage. Finally, fibrocartilage. In fibrocartilage, the ECM contains dense type 1 collagen fibers, making it more acidophilic. The isogenous aggregates are aligned, and fibrocartilage does not have a perichondrium. Where do we find fibrocartilage? Examples include the pubic symphysis, articular discs in the sternoclavicular and temporomandibular joints, and the menisci. And that's it. That wraps up the key points about cartilage. I hope you found this interesting.